Hello students, Mrs. Kleins here with your next section of video notes over protein synthesis, also known as the central dogma of biology. This is perhaps one of the most important concepts or principles that we learn about in class. So, before we jump into protein synthesis, we need to spend a little bit of time highlighting the major structural differences between DNA and RNA. We're familiar at this point with DNA. It's a double-stranded or double helix molecule. The sugar that makes up its nucleotide structure is deoxyribose. It's got 4 different nucleotides based on the bases that the nucleotide contains, meaning some nucleotides will have adenine, some guanine, some thymine, and some cytosine. And we remember in the DNA structure how they form base pairs, purines and pyrimidines, for forming base pairs together as determined by Chargaff's rule. We know that DNA has to be found safely in the nucleus. It's a very large molecule and it can't easily leave the nucleus. It is kept there nice and safe. It serves as the code for building proteins, so every single one of your body cells has an identical set of DNA. And we learned how DNA gets copied in the last section of notes during DNA replication. So, each new daughter cell that divides receives a full set. RNA is a little bit different in that it is a single-stranded molecule. It is also made up of nucleotides, but the sugar in the nucleotide is ribose. And the 4 different nucleotides have a lot of similarities, but there's one major difference to note. RNA will not contain thymine in its code. It will have uracil, usually abbreviated with the letter U, but like thymine, it'll form base pairs with adenine, and we'll talk more about that soon. RNA can be found throughout the cell. It's not limited to stay in the nucleus. However, some processes that involve RNA do occur in the nucleus, but then others will take place in the cytoplasm of the cell. The function of RNA is that it carries the code from the DNA to other parts of the cell and aids in protein synthesis. So it essentially is like the transport molecule of DNA. There are 3 different types of RNA that you should be familiar with, and we will discuss those soon. Here's a slide showing you some slight differences between the DNA structure and the RNA structure. Here are your 3 different types of RNA. mRNA, which stands for messenger RNA. Messenger RNA carries genetic information from the DNA sequence out of the nucleus to the cytoplasm. rRNA, or ribosomal RNA, is what makes up ribosomes. You should be familiar with those because they are the organelles that make proteins. They almost look like these little hamburger buns. There's 2 different units to an rRNA structure, or ribosomal structure. There's a larger top subunit and a smaller lower subunit, and we'll talk soon how mRNA locks into rRNA to make proteins. tRNA is called transfer RNA. Transfer RNA brings amino acids to the ribosome for protein synthesis. So it transfers amino acids that bind to the bottom of the tRNA structure out of the ribosome for protein synthesis. So the goal of protein synthesis is to express the genetic code in the form of proteins. Remember that DNA is code. It's like a recipe book, but beyond that, it doesn't do much for the cell. It gives the code or instructions for making all of the proteins that serve the cell, that make up the cell, that carry out important functions within the cell. So proteins are the major structural and functional components of our bodies and cells. There are 2 main steps to protein synthesis. The first one we'll talk about is transcription. And these steps, again, they occur during interphase of the cell cycle because that's when the cell is kind of growing. It's growing, it's carrying out daily processes, it's where the cell spends most of its life. So it's making the proteins and organelles it needs to function and to maintain homeostasis. So transcription occurs in the nucleus of the cell, and translation will occur in the cytoplasm using ribosomes or rRNA. Again, I just wanted to emphasize the importance of DNA being a code. Each strand of your DNA in the nucleus bundles into a structure that we'll talk more about called a chromosome. Each one of these little chromosomes that you see here on this picture on the bottom left is made up of one strand of DNA that is very tightly coiled. You have 46 chromosomes in every cell in your body, with the exception of reproductive cells, which have half the number of chromosomes. In every single strand of DNA, there are up to 250 million base pairs in very small sections of these chromosomes that code for different genes. Remember, genes are going to be the traits that you inherit from your parents. Each gene contains genetic code to produce one specific protein. So during transcription, one small segment for one particular gene gets transcribed and then later translated into a protein. Let's start there. Transcription. If you've ever heard of a profession called transcription, it's where you take and write down notes, either things that people are speaking or things that are heard in a specific setting, so transcribing is kind of making a copy or documenting something. The process of transcription is kind of like that. It occurs in the nucleus, and what happens during this process is a small section for one specific gene located on DNA is copied into mRNA. mRNA is going to be a very small structure compared to the entire length of the DNA strand. And it may just be one small gene that gets copied into mRNA from the original DNA sequence. So this image on the right is chromosome 15, just to show you. And there are over 100 million base pairs just on this one chromosome that code for all of these different genes. There are major steps in transcription. The first step is what we see in DNA replication. The first step is like what we see in DNA replication. A small region or section in the DNA that codes for a specific gene will begin to unwind. The hydrogen bonds will break between bases using the same helicase enzyme. This is more of a temporary process, though. We're not going to make copies of the entire strand of DNA, but just a small segment of one specific gene. RNA polymerase is going to be the next enzyme, and what it does is it's going to copy that small section of DNA by adding complementary RNA nucleotides. So it's going to be A, C, G, or U nucleotides that are added now, complementing the parent strand. After the mRNA sequence is complete, it will leave the nucleus and it will travel through the cytoplasm to a ribosome, but the base pairs will form back between the DNA strands and the DNA remains safely in the nucleus. So here's just a picture showing you that. You've got a small section of DNA that's been unwound by helicase, and the RNA polymerase is forming fake complementary bases to that DNA template just in one small section that codes for one specific gene. Now this mRNA strand is going to be pretty small, so it can leave through the nuclear pores in the membrane of the nucleus. Let's kind of show you how that process works. You've got an example. This is a double-stranded helix DNA sequence, and you know that because it contains thymine. And remember that DNA strands run anti-parallel, meaning they run in opposite directions. That's what this 5' and 3' designates the orientation of the phosphates and sugars in the backbone of DNA. So in transcription, it's important that you know that one strand of DNA is called the coding strand, and this is because we see it's going 5' to 3'. This is going to be very, very similar to our mRNA sequence that you transcribe. The main difference you'll notice is that RNA will not have thymine. The other strand is called the template strand of DNA, and this runs from 3' to 5'. This is used as a template to build the messenger RNA. It's important whenever you're giving information on any activity or an assessment to note that if it tells you the coding strand, that tells you something about the mRNA sequence. If you're provided the template strand, you know that the template strand is used to build or form complementary bases to make the messenger RNA or the mRNA. I have these bases broken out into these 3-piece segments called codons, and we'll talk more about codons in a bit. But a codon, by definition, is a 3-nucleotide-based sequence. So whenever your ribosomes read the messenger RNA, they're going to read them in these 3 base segments called codons. Finally, you're going to use a process very similar to what you did in DNA replication to make your mRNA sequence or messenger RNA. So you're going to form complementary bases using the template strand to form your messenger RNA. So thymine will bind to adenine, adenine will bind to uracil, and cytosine will bind to guanine. There's an mRNA sequence that's going to also run 5' to 3'. Those are going to be your complementary bases, and your coding strand of DNA is almost identical to your messenger RNA with one exception. So you should see that in the next codon, but even if you didn't have the coding strand to look at, you would know that cytosine bonds to guanine. You've got guanine, then cytosine. Your next codon, you won't have any thymine, but you'll have uracil. And finally, you've got this last codon, UGA, which we'll later learn is going to be a stop codon for protein synthesis. So once you've got your complete set of messenger RNA transcribed using the template strand of DNA, the mRNA strand will leave the nucleus and will be ready for the next process called translation. So translation occurs in the cytoplasm, and it will happen on a ribosome, which remember, your ribosomes are made of rRNA. The goal of translation is that mRNA sequence, which was almost used like a photocopy of a gene, is used to make a protein from a chain of amino acids. Proteins, remember, are a macromolecule made up of amino acid monomers. So the steps of translation look like this. First, the messenger RNA that was made in the nucleus attaches to a ribosome, and, as I said earlier, it kind of looks like a hamburger bun. The upper and lower subunits will lock onto the messenger RNA, and then the ribosome will begin reading that messenger RNA by codons, or groups of 3 bases, until it reaches what we would call a start codon. Once it reaches that start codon, the transfer RNA, which contains anticodons that will be complementary to the codons on the messenger RNA. The tRNA will temporarily bind to the codon and will bring an amino acid with it. So the amino acids get dropped off by the transfer RNA at the ribosome based on the specific mRNA sequence. So transfer RNA, there are 64 different codon combinations and there are 20 different amino acids. Those anticodons are really important for identifying when they bind to the codon which amino acid needs to be brought in order for protein synthesis. This process continues in the ribosome until the stop codon is read. There are 3 different stop codons in the mRNA sequence, and essentially that will stop protein synthesis and allow the messenger RNA to be released from the ribosome and the protein to continue folding and bonding so that it can carry out its important function in the cell. Important vocabulary: peptide bonds are the bonds that hold the individual amino acids together. So remember, they get brought into the ribosome by the transfer RNA, and as they get dropped off one by one, the amino acids are chemically bonded using peptide bonds to form a polypeptide or a protein structure. So these are some terms from earlier units we've covered. And just remember, as I stated earlier, amino acids are the monomers of proteins. So this is what the process looks like. You've got your ribosomal RNA. Your ribosome, again, it kind of looks like a hamburger bun. You've got your messenger RNA depicted by the pink strand. So this, again, was made during transcription in the nucleus of the cell. And so it will lock into the upper and lower subunits of the ribosome, and it clicks one codon at a time through the ribosome. The bluish little guys floating around represent your transfer RNA. They carry the amino acids, so you can see the one with GLY attached as an anticodon that complements the messenger RNA codons where the arrow is pointing. So it's going to be G to C, G to C, C to G. And when it binds, it brings the amino acid attached to it. GLY is the amino acid attached here, and then it will form a peptide bond between PHE and GLY, and then it's going to add to the growing chain of polypeptides, which will eventually fold and make a protein. So this process is translation, taking this code and making it meaningful to the cell. To understand which amino acid gets brought to you, you can use the mRNA sequence right here and a codon chart. In some cases, you'll be asked to translate a sequence of messenger RNA into an amino acid sequence. There are 2 different codon charts. You don't have to memorize these, but I do want you to be familiar on how to use both of them. In this codon chart on the left, when you're looking at a codon, a 3-base sequence, you're going to start in the middle. The first base you'll find in the middle. So let's say that we're going back to this example here. I'm looking at GGC. So G is my first base, G is my second base, and C is my third. So I go to the middle, go G, G, C. So that codes for glycine, GLY. You can see that some amino acids will have multiple codons that code for them. Like in this case, there's multiple codons that would give you glycine. Using this chart on the right, you may receive this chart on a test or an assessment, so that's why you need to know how to use both. You should get the same results. So you've got G for your first base. So I'm going to look at the bottom region for my first base. My second base is G, so I'm going to be looking in this bottom right section. I'm looking at my third, which is C, which is on this axis. So I had G, which brought me down, G, which brought me over here, and then my third letter is C. So it gives me GLY, which is the same as glycine. So what you're going to do next is you've got a messenger RNA sequence that's been provided below. You've got the 5' to 3'. So you're going to start here with the codon AUG. So I'll start by using this chart. A is my first base, right? So that'll take me right here. U is my second, so I move into this section. And then G is my last letter. That gives me MET. Okay, if I go over to this side, same thing. I'm still doing AUG. So I start in the middle and I work my way out. So then I've got A, U in the second level, G in the third, gives me MET or methionine. Good. So then that's my first amino acid. CGC is my next codon. So if I look at it, starting in the middle, I've got C, G, C. That gives me ARG. And you can abbreviate them with the first three letters of your amino acid sequence. Then my next one is UUU. So if I start on this chart, I've got U for my first, U for my second. So I'm in here. U for my third. That gives me PHE. And my last one is UGA. Start in the middle. UGA. Stop. That is going to signal that protein synthesis is complete. So all amino acid sequences will always start with MET. That is essentially called the start codon. We can see that there are 3 different stop codons, but all of your amino acid sequences will always start with the MET amino acid.